Hello everyone. Today I will introduce a research article published in iMetaOmics, entitled Intact Pulse-Cotyledon Cell Diet Improves Systemic Growth via the Somatotropic Axis in Malnourished Mice. Malnutrition remains a major global health challenge, affecting more than 10% of the world's population, especially in low-income regions such as East Africa and South Asia. In 2022 alone, about 149 million children under 5 were stunted, and 45 million were wasted. Malnutrition impairs growth but also causes long-term economic, social, and health consequences. Recent studies show that malnourished children often exhibit immature gut microbiota with reduced diversity. The establishment and maturation of the gut microbiome in early life are critical for nutrient absorption and healthy growth. Therefore, the gut microbiota has become an important therapeutic target for the prevention and treatment of malnutrition. Among microbiota-directed diets, pulses are considered key ingredients. Pulses are rich in dietary fiber and resistant starch. Intact pulse-cotyledon cells retain their cell wall structure and act as resistant starch type 1, allowing nutrients to reach the colon and be fermented by gut microbes. In contrast, pulse flour disrupts cellular integrity, leading to rapid digestion in the upper gastrointestinal tract. Based on this, we hypothesized that intact pulse-cotyledon cells may provide greater benefits than conventional pulse flour in alleviating malnutrition. To test this hypothesis, we performed a comparative study in a mouse model of malnutrition. First, in terms of systemic growth, malnourished mice showed significantly reduced body weight and length compared with controls. After 4 weeks of intervention, the intact cotyledon cell diet significantly improved body weight gain and feed-con efficiency, with stronger effects than the pulse flour diet. At the tissue and organ level, malnutrition reduced bone density, bone area, adipose mass, and liver weight. The intact cotyledon cell diet significantly improved bone area, increased adipose mass, restored liver weight, and alleviated hepatic steatosis and inflammation. Regarding metabolism and endocrine responses, the intact cotyledon cell diet restored glucose tolerance, improved lipid metabolism, and reduced systemic inflammation. It also significantly increased serum levels of growth hormone, IGF-1, and IGF-BP3, indicating enhanced growth-related hormone secretion. In the gut microbiota, the intact cotyledon cell diet increased microbial diversity and enriched beneficial carbohydrate-utilizing bacteria, particularly lactobacillus, which are associated with healthy growth. Metabolomic analysis showed that the metabolic profile of the intact cotyledon cell group was closer to that of the healthy control group. Differential metabolites were mainly involved in amino acid metabolism, glutathione metabolism, and steroid hormone biosynthesis, indicating a reshaping of the intestinal metabolic environment. Short-chain fatty acids were significantly reduced in malnourished mice, whereas the intact cotyledon cell diet markedly increased acetate, propionate, and butyrate levels. Correlation analysis showed that lactobacillus abundance and SCFA levels were positively associated with body weight, adipose mass, bone density (GH) and IGF-1 levels. Mechanistic analysis of the somatotropic axis revealed that the intact cotyledon cell diet increased IGF-1 and IGF-BP3 expression in the liver and peripheral tissues, upregulated GHR expression, and restored hepatic FFAR2 levels. These findings suggest that SCFAs derived from intact cells activate the somatotropic axis through the gut-liver axis, thereby promoting systemic growth. In conclusion, intact pulse-cotyledon cells are more effective than conventional pulse flour in alleviating malnutrition. This effect is mediated by enrichment of beneficial carbohydrate-utilizing bacteria, enhanced SCFA production, activation of hepatic FFAR2, and stimulation of the somatotropic axis. This study highlights the importance of preserving cellular integrity in pulses and provides new insights into whole food-based strategies for combating malnutrition. Thank you for your attention. You are welcome to download and read the full article, and please feel free to contact the corresponding author if you have any questions.